Welcome to the Lavaux Vineyards. This is where our project began. Isn't it pretty? Well, don't get too excited. There's a very serious and ongoing problem here. When we began our journey with iGEM, we went looking for a local issue that we could work on. These vineyards seemed like a great place to start, as they are close to our university and the prize jewel of our region. It was here that we found one of the biggest problems in the region: copper pollution. So here's what's going on. Copper-based fungicides are widely used in vineyards to protect the plants from a disease called mildew, among others. The fungicide is sprayed onto the leaves, allowing the copper to form a protective layer. The treatment is effective, but unfortunately, the copper solution has to be reapplied every time it rains. The copper ionizes in the rainwater, thus dissolving the protective layer. As a result, copper accumulates in the ground. Copper being a heavy metal, such high concentrations wreak havoc in the topsoil ecosystem. Interestingly, the aforementioned copper fungicides are by far the most effective organic treatments available on the market to combat mildew. Thus, if a winemaker chooses to abide by the organic label, they are condemned to use this fungicide, thus contaminating their own soils. This practice is also very common across the world in other leafy-type plantations, including but not limited to tomatoes, basil, and tea. That's right. Those organic labeled products in your shopping basket are not all that harmless to the environment. Throughout this video, we will take you on our iGEM journey to save the Lavaux Vineyards, combining solutions in chemistry, synthetic biology, electronics, and computer science. In a nutshell, we decided to collect the copper-contaminated rainwater, channel it to a prototype bioreactor, and treat it with genetically modified yeast capable of fixing copper. From the very start, it was evident to us that for such a project, we needed to engage closely with the communities affected, as well as with various other relevant stakeholders. That is why we will continuously refer back to the stakeholders we have met to show the impact they had on our project. Moreover, through our work in education and communication, we attempted, to the best of our abilities, to shed light on this problem, as well as on the potential of synthetic biology. Now, let us tell you about our work.
We began our project by visiting local winemakers to ask them about their opinions and concerns about copper pollution. They confirmed to us that they needed a solution to this problem. So we took advantage of the situation and pitched a few of our first ideas for implementation. It was crucial for us to hear their opinion, as preserving their economic livelihood was a key goal of ours. Out of all the implementation ideas we proposed, the one that generated the most interest was our simple carpet design. This design, laid out under the vine ranks, would catch the copper-contaminated rainwater, which would then be brought to a bioreactor. One winemaker highlighted that the carpet design would block the growth of weeds under the vine ranks, thus decreasing labor and making this implementation more economically viable. Now that we had a way to gather the rainwater, we wanted to know exactly what copper concentrations we were going to be working with. So we took a few field trips to the fields. Field trips to the fields? Who wrote this script? We gathered over 80 samples of soil, rainwater, and leaves to better understand the challenges we were to be facing. Our conclusions were the following. Firstly, copper concentrations in the soil were very high, going up to 160 milligrams of copper per kilogram of soil, even though the soil is considered as polluted from 40 milligrams per kilogram. Secondly, after doing tests replicating rainfall on vine leaves, we determined that the copper seems to be completely washed off, as from 4 millimeters of rain. Lastly, the concentration in the rainwater seems to be around 2 milligrams per liter. With this information in mind, we could start designing our synthetic biology solution. Now bear with us while we tell you about the more technical stuff.
Now that we had a way of catching the copper-contaminated rainwater, we needed to treat it. So we came up with an idea of using a microorganism capable of absorbing copper in water. The initial goal was to assemble a complex able to accumulate copper both inside and outside the cell. Here are the three main components. Number 1, an intracellular protein for cytoplasmic storage of copper ions. Number 2, a transmembrane protein allowing ions to enter the cell from the external environment. Number 3, a metallothionein expressed on the extracellular surface to bind ions. However, we realized that the first two functions of this complex are already endogenous to the wild-type yeast, that is, transporting the copper ions into the intracellular space and store them there. Nevertheless, why don't we try expressing the same intracellular protein responsible for binding copper ions on the cell surface? Oh, this isn't an in-person presentation? The protein we chose is called copper metallothionein 1,1, and it's encoded by a gene called CUP1. This protein naturally contains 4 different binding sites for divalent metal ions and 8 for monovalent ions, with very high affinity for copper and cadmium. To improve the capacity of the cell to retrieve copper, we thought of expressing the copper intracellular yeast metallothionein CUP1 at the surface of the cell. The first step in this process was to clone its coding sequence. We chose the PCTCon2V5 plasmid. This plasmid allowed us to fuse the CUP1 protein to a yeast surface display system consisting of AGA2, a protein that links by disulfide bonds, to AGA1, a yeast membrane protein. We first checked the protein expression by Western blot using a primary antibody targeting a V5 tag. Then we performed immunocytochemistry to visually check the presence of CUP1 on the outer membrane of our cells. To improve the copper binding efficiency, we designed CUP1 dimers, 2 copies of CUP1 fused together by a linker, and completed the same workflow as for the monomer. We designed the linkers and modeled them in AlphaFold to obtain the most stable and efficient structure. To avoid any problems of homology, we decided to use 2 slightly different CUP1 sequences. Furthermore, to maximize the yield of the protein production in yeast and prevent self-hybridization of the RNA after transcription, the full nucleotide sequences of the CUP1 dimers were codon optimized. We observed excellent results in both expression and cloning for each dimers and for the monomer. We hypothesized that doubling the CUP1 presence on the cell surface should ideally lead to a 2-fold improvement of copper uptake and resistance. Another question we raised was, does our expression system affect the growth of yeast in any way? The wild-type EB100 yeast normally only grows in a complete yeast medium, such as YPD, which is yeast extract peptone dextrose, whereas the yeast transformed with PCTCon2V5 vector were selected in SGCAA, which is a reduced medium containing galactose but lacking tryptophan that was used for auxotrophic selection of yeast transformants. For that purpose, we monitored the growth over time of the transformed yeasts in both rich media, YPD, and selection media, SGCAA, as well as the growth of wild-type EB100 yeast in rich media by measuring the culture's OD600 every 15 minutes. With no surprise, the transformed yeasts with the backbone vector grew faster in rich media, similarly to wild-type yeasts. It took longer for the engineered yeasts, transformed with backbone vector or CUP1 construct, to grow in selection media, but the growth curves still reach a plateau. Moreover, we also performed growth assays with the wild-type EB100 yeast in YPD media, supplemented with various concentrations of copper. Our yeast strain demonstrated good resistance to growing concentrations of copper, up to 1,000 mg per liter, validating our choice to use it as a chassis for copper remediation. To conclude our lab work, we performed tests monitoring yeast ability to remove copper in different conditions compared to wild-type yeast. So, how did we measure our microorganisms' ability to remove copper from a solution? We monitored the copper concentration of the medium in contact with yeast over time. The conditions varied between yeast strain and yeast concentrations. We started by collecting samples from the cultures at different time points. We then spin the cells down by centrifugation, and finally we measured the copper concentration in the supernatant using a colorimetry test with cuprizone and a spectrophotometer. The first strain we tested was the wild-type yeast. We found out it was effectively removing copper from the solution. Additionally, the denser the yeast population, the greater the ability to fix and remove copper. We next tested our engineered yeasts. In a first batch of experiments, we compared the CUP1 monomer and 3 different CUP1 dimers' ability to remove copper compared with the wild-type strain. Unfortunately for us, we observed that our modified strains, CUP1 monomer or CUP1 dimer, were not only not performing better than the wild type, but did not even absorb copper in solution at all. Disappointed by this finding, but not less determined to find out why, we continued digging. 3 variables changed from wild type to engineered yeasts: the membrane expression of CUP1, the medium, and the fact that the yeast is genetically modified or not. We therefore had 3 different hypotheses. Number 1. The extracellular membrane expression of CUP1 interferes with the endogenous bioaccumulation property of yeast by, for example, blocking the entry of copper into the cell. Number 2. Transformation of yeast with PCT-CON2V5 vector, with or without CUP1 insert, interferes with the endogenous bioaccumulation property of yeast. Number 3. The reduced medium is the problem and prevents the yeast from bioaccumulating copper. Neither the backbone system in SGCAA nor in SDCAA was retrieving copper from the solution. As the system backbone in SDCAA was not expressing the surface display, we could eliminate the first hypothesis. Indeed, the only differences between the wild type and backbone system strains were in the medium, and in the fact that the backbone system had been genetically modified. Therefore, we decided to check if the problem was coming from the medium by testing our engineered yeast in YPD plus galactose to induce the system, as well as the wild-type strain in SGCAA plus tryptophan, because it would not survive without tryptophan. Seeing that the wild-type yeast in SGCAA supplemented with tryptophan was behaving like the engineered version in SDCAA, and that engineered yeast in YPD supplemented with galactose was behaving like wild type in YPD, made us eventually conclude that the problem was, in fact, coming from the medium. Ultimately, we found that our engineered yeast, even when cultured in YPD, was not better at absorbing copper than the wild type. To conclude, our strategy based on CUP1 surface expression did not improve the endogenous bioaccumulation ability of yeast. Now that you have a complete overview of our synthetic biology solution, let's check in with the hardware.
Welcome to our electronics lab. This is where the implementation came to life. We showed you how we planned on capturing the copper-contaminated rainwater, and how we planned on treating it using modified yeast strains. However, since it's illegal to release GMOs into the environment in Switzerland, we needed to isolate them in a bioreactor. First, we wanted to build a flow-through reactor. Unfortunately, simulations showed that the water had to be flowing fast. Too fast for the biological solution. So we designed a simple, static bioreactor. As water enters, we measure its volume thanks to a pressure sensor to see if the container is full. Inside the reactor, yeast fixes the copper in the water for a set amount of time. The yeast cells, in turn, are fixed onto alginate beads. The beads' large size allow us to filter the water using a simple grid filter. This idea came up after a discussion with various experts from local water treatment facilities. After the treatment time is up, a solenoid valve lets the water out. We decided to add electronics into our solution, as we wanted an autonomous system to avoid additional labor. However, the idea of a recipient containing yeast cultures fixed onto beads could be much more generally applicable. For example, in areas where manual labor is still the most common practice, one could forget all of the electronics, thus making our solution much cheaper and more adapted to traditional agriculture.
Before you click away, as stated in the beginning of this video, education and communication were very important to us. Here are a few of the projects we developed during the summer. We are very proud of our educational children's book, telling the story of a village and their struggle to combat mildew. We also made a podcast on GMOs for the general public, hosting interviews with different experts. It is in French and available now on Spotify. Furthermore, we visited high schools across the region, teaching the students about synthetic biology, and organized a high school lab immersion here at EPFL. Finally, we hosted an iGEM recruitment event to encourage EPFL students to join next year's team. When this project began, we were not aware of copper contamination in organic agriculture. It was only by engaging with relevant stakeholders that we were able to grasp the magnitude of this issue, as well as how it affected our local area. Although not perfect, we presented a proof of concept to solve the issue of copper contamination in organic vineyards. We should all be concerned with the welfare of our environment. Thus, we hope to have raised awareness on the impact we can have, even in cases such as organic agriculture. Add more script.
Hello?
Add credits.
Hello?
Add
No, we're done.
What?
We're done. That's the end of the script.
Oh, sorry. The wiki freeze really took a toll. EPFL team,
Signing out.